Okay, welcome back everybody. We are going to start the heart today. So the heart is a four-chamber muscular pump, and it represents the center of the cardiovascular system. Now, although we say it's a four-chamber muscular pump, I kind of look at it and say, well, it's two pumps in one. And when I say two pumps in one, we're going to look at the right and the left side of the heart. Now, remember, we're always looking at this in anatomical position, so the person is facing you, which is why that's left and that's right. So this side of the heart, the right side, is one type of pump. And then the other side, the left side, is another pump. So it's definitely four chambers, right? 1, 2, 3, 4 chambers, where you have the upper chambers being the atrium and the lower chambers being the ventricles, but then two pumps in one. This right side is going to be considered pulmonic, whereas the left side is systemic. And we'll get into a little bit more detail as to what we mean. The term pulmonic is obviously referring to the lungs, and systemic is the heart has to find a way to pump that blood systemically to all the other systems in the body. But pulmonic has to find a way, the right side has to find a way of pumping the deoxygenated blood to the lungs to pick up oxygen. Left side is going to take the oxygenated blood and pump it to every other part of the body. So the heart's about the size of your fist, and it's located in a region called the mediastinum, which is superior to the diaphragm. There is some attachment from the diaphragm to the inferior apex of the heart. It's to the left of the midline. It's anterior to your vertebral column, but it's posterior to the sternum. So that region is referred to as the mediastinum. And we can see it here, and we can also see it here. You could see a few landmarks. Let me just change the color of the pen. Let's change it to blue. So up on the top left there, you can see the diaphragm. And we can see the sternum. A really important landmark is the second rib. And when you review some of your bony anatomy and you look at the sternum, there's like this top part of it that looks like this, the manubrium, and then it kind of goes like this, and then it kind of tapers off to the xiphoid like that. But this region right here, let's make this, let's say this is a little bit bigger. Let's make it like that, okay? So this region here between the manubrium and then the body of the sternum, right there, I'll make an arrow. This section here is the sternal angle. And everyone has this little bump. If you were to feel this section here, the jugular notch, which is easy to palpate, which is right there, or the suprasternal notch, when you find that, and then you go down, let's say 1, 2, 3, about 3 to 4 finger breaths, is where you get to that sternal angle or angle of Louis. And that's always here at the level of the second rib. And the reason that's important is once you find that second rib, then here's the second intercostal space. And then here's the third rib, here's the third intercostal space. Then here's the fourth rib, and then there's the fourth intercostal space. And being able to know that is what tells doctors where to place the diaphragm or the bell of the stethoscope when they're listening for heart sounds. They just don't place it anywhere. I mean, if they place it over here, they're going to be placing over bone. They want to be able to place it in the hollowed out section so they can hear the echo of the heart sounds. Again, another good picture of the mediastinum. Really nice picture because this illustration here shows a few things. Now, some of the things I want you to pay attention to is the color, right? So if you look at the right atrium and the right ventricle, you'll see that what we have here and here is blue. What we have moving up in this direction is blue. What's moving in this direction and this direction is also blue. Why? It's deoxygenated. The blood that's there doesn't contain oxygen, so it's deoxygenated blood. The vessels, if you look at the direction going from the right atrium and blood going down to the right ventricle and then pumping up into the pulmonary trunk, into the pulmonary arteries, that's deoxygenated blood. Remember, arteries carry blood away from the heart. Veins bring blood to the heart. So it's a little bit tricky when you look at the colors and the direction, because most of the time you'll think, well, blood going away from the heart is going to be arterial, and arteries are carrying oxygenated blood, but that's not the case here. So right here on top, this is the superior vena cava, and down below, coming up this way, that's the inferior vena cava. And the superior vena cava is bringing blood from the head and the upper extremities into the right atrium, and the inferior vena cava is bringing blood from the abdominal section, the lower extremity, bringing that deoxygenated blood into the right atrium. Okay? So right side is pulmonic because you can see that the right atrium and right ventricle is responsible for pumping blood into the pulmonary trunk, into the pulmonary arteries, and the pulmonary arteries drive that deoxygenated blood into the lungs, and then the pulmonary veins are going to pump oxygenated blood into the left side of the heart, into the left atrium, down into the left ventricle, and then into the aortic arch. Okay, there's going to be a video that I'm going to show of me just drawing out the blood flow through the heart. So let's take a look at that. I think that may be coming up here. Let's see if we could take a look.
Hi, welcome
Okay,
back.
so let's see if I can make this a little bit larger for you. Let's see here.
For our anatomy lab. So
Okay.
let's take a look at the heart. I'm going to draw out a very simple picture. And I make the heart this way for a very particular reason. If we look at how I'm drawing this, you can see that I'm intentionally making the left side of the heart larger and thicker than the right side of the heart. So the myocardium, this muscular layer of the right side, is a lot thicker than the left side because the left side of the heart itself is like two pumps in one. The left side dealing with systemic circulation, while your right side is dealing with pulmonary circulation, meaning the only job the right side has is to pump blood to the lungs to pick up oxygen. While the left side of the heart, it's taking that oxygenated blood and now pumping it to all the systems of the body. So let's take a look at some of the structures here. We're going to have blood that gets into the heart, into the right atrium, by way of the superior vena cava. And also blood comes from the lower extremity into the right atrium through the inferior vena cava. Again, deoxygenated blood, red blood cells that don't carry oxygen are going to be pulled into the right atrium. The pressure builds up in that right atrium, and this valve is going to open up to allow blood to flow into your left ventricle. I'm sorry, your right ventricle. So from the right atrium, blood flows into the right ventricle by flowing through this valve. That valve is referred to as the tricuspid valve. 3 cusps to it. So now we have pressure in the right ventricle that builds up, and when the pressure in the ventricles build up, it will pump the blood from inferior to superior. From the atriums, blood flew from the atriums into the ventricles from superior to inferior, but from the ventricles, blood will pump up from inferior to superior. And that blood is going to go through a valve here that's called the pulmonary semilunar. As the blood pumps up, this valve is now forced shut. That's what gives you the lub sound, lub-dub, lub-dub. It's the closure of that tricuspid valve. As that valve shuts, this valve opens, and blood is now pushed upward through the pulmonary trunk. After the pulmonary trunk, blood is diverted into your right and left pulmonary artery, which will go into the lungs, and a right pulmonary artery, which also goes into the lungs. So blood is pushed up through the pulmonary trunk into both pulmonary arteries, a right and left pulmonary artery, into the lungs. So that's the lungs. Oxygen is picked up. After the oxygen is picked up, blood is now forced into the left atrium from both the right lung and the left lung. So let's make these vessels. I'm making 2 from here and 2 from here. These are now called the left pulmonary veins and the right pulmonary veins. On the models, you will notice that the pulmonary veins appear red because they are now carrying oxygenated blood. On the model, you'll notice that the pulmonary trunk is blue and the pulmonary arteries are blue because they are carrying deoxygenated blood. So now we've got pulmonary veins that are pooling oxygenated blood into the left atrium. When the pressure builds up in the atrium, this valve opens and the blood will dump into your left ventricle. Again, when that blood in the ventricles start to fill up, it will also pump blood from inferior to superior, pumping blood up this way, inferior to superior, through this valve. So as that blood is pushed upward, this valve is now closed. When this is forced closed, your semilunar opens. This semilunar is called the aortic semilunar. So that's the aortic semilunar. On the other side was the pulmonary semilunar. So this comes up. Let's follow that up into the aorta and the arch of the aorta. So here's the aorta. This is the arch of the aorta. And off the arch of the aorta, we have 3 blood vessels. I'm going to make 1, obviously going to the right side, and we'll make 2 going more to the left. The one that goes to the right is the brachiocephalic, and the 2 to the left, this one here being the left common carotid and the left subclavian artery. This will then continue down into the thoracic aorta, all the way down into the abdominal aorta, and then that will split, going to the lower extremities, into the common iliacs that will split into an internal and external common iliac that will feed the entire lower extremity. I will also point out, and I'll go over this with the models with you, that in the ventricles we have myocardium, we have muscle, rough muscle down in here called trabecular carnae. You have them in both the right and left ventricles, trabecular carnae. And off these trabecular carnae, there are these little mounds or little hills. And those mounds and hills are called papillary muscles. And these papillary muscles are going to attach to these valves by way of tendons called Chordae Tendinae. So these Chordae Tendinae muscles attach to these valves and prevent them from overclosing into an open position, which can potentially cause a murmur. Okay? The muscles that you find in the atrium, these are called pectinate muscles. In the ventricles, they're called trabecular carnae. Okay, please stay tuned, and you will look forward to looking at
one of the
these
things that I
structures now
wanted
in the heart.
to mention to you here was that over here, off the abdominal aorta, there is the celiac trunk, and then there's the superior and inferior mesenteric arteries, and then there's the gonadal arteries, and then the renal arteries. So the abdominal aorta has primary blood vessels coming off of them that are going to bring oxygenated blood to the main organs. Off that celiac trunk, there's the common hepatic that's going to bring blood to the liver. There's a splenic artery going to bring blood to the spleen. And off the celiac trunk, there's a left gastric artery that's going to bring blood to that lesser curvature of the stomach. And if there's a left gastric artery, there's going to be a right gastric artery. That's going to come off, that's a branch off the common hepatic. Then, as you move a little bit further down the abdominal aorta, there's another vessel called the superior mesenteric, which is going to hit a majority of the remainder of the digestive tract. Small intestines, duodenum, jejunum, and ileum, the ascending colon, and about half of the transverse colon. That's all superior mesenteric artery. And then the remaining of the GI tract, like the other half of the transverse colon, the descending colon, the sigmoid colon, the rectum and anus, all that's going to be the inferior mesenteric artery. And then you have the gonadal arteries that come off, and then the lumbar artery. So there's many, many blood vessels coming off of that abdominal aorta, not just splitting into the common iliacs going to the muscles, but also to the organs. Okay? All right, so hopefully that was helpful for you. And so this diagram here, for those that are more linear, I think it's important to be able to draw this out. Okay, you want to start, and you want to start with, the right atrium, and then next to it, you'd want to draw heart, and I would just visualize what's happening as you do this. And start in the right atrium, right? Make a number 1 and just work your way down. From the right atrium, it's going to go through the tricuspid valve into the right ventricle. So it's going to go this way. And then from the right ventricle, it's going to go through the pulmonary semilunar valve. So that pulmonary semilunar valve is going to be here. And then from that, that's going to come up into the pulmonary trunk, and that's going to split into the pulmonary arteries, going into the lungs. Okay? Then, from the lungs, pulmonary veins. Now remember, the pulmonary veins are going to be red because they're carrying oxygen back into the left atrium. All right. So now they're into the left atrium, through the bicuspid valve into the left ventricle. So it's going to bring blood down this way. Then from the left ventricle into the aortic semilunar valve. So that aortic semilunar valve is going to go here. And that's going to lead into the aorta, in the arch of the aorta, where we then have a brachiocephalic artery, a common carotid on the left, and a left subclavian. Right? If we draw a line there, then this is right. So the brachiocephalic is on the right, and then you have the two vessels on the left. This one here being the left common carotid, this one here being the left subclavian artery. Okay, and then now you have blood from this vessel here, brachiocephalic, the brachial division is going to bring blood to the arm. The cephalic, cephalad means head, it's going to bring blood up to the head. On the left side, we have this one here, which is the common carotid. The left common carotid is going to bring blood to the brain. And then you have the left subclavian, that's going to bring blood down to the arm. So both on the left side and the right side, you got blood vessels bringing oxygenated blood to the brain and the arm. It's just the names of the vessels change. And then, of course, it's going to come straight down through the thoracic region into the abdominal aorta and then down the legs, just like the video showed. Okay. All right, let's look at some of the cardiac muscle anatomy overview. I think this just may be an opportunity for you to kind of look for clicking on things. So here's the tricuspid valve, the aortic semilunar, bicuspid or mitral valve, pulmonary semilunar, the myocardium, there's the arch of the aorta, left common carotid, superior vena cava, pulmonary trunk, left pulmonary artery, left pulmonary veins, right pulmonary artery, right pulmonary veins, right atrium, right ventricle, left atrium, left ventricle, the IVC, and the coronary arteries. Okay, there's a left and a right coronary artery. The walls of the heart, epicardium, myocardium, and endocardium. 3 layers, right? Always loves the 3 layers. Major organs, you know, if you remember when we did reproductive and we looked at the smooth muscle of the uterus, there was the endometrium, the myometrium, and the perimetrium, right? When you look at muscles and we look at muscles and then we look at the nerve, things always have layers, all right? Protective sheets, protective coverings. So here on the upper right, you can see here's the epicardium, there's the myocardium, and there's the endocardium. Remember, 3 types of muscle in the body: smooth muscle, skeletal muscle, and cha-ching, cardiac muscle. So the pericardium, the heart is surrounded by a group of membranes, collectively referred to as the pericardium. And the pericardium is going to have these 2 primary layers. So there's a fibrous pericardium, and then there's a serous pericardium. So when we look here, here is the fibrous pericardium, and here is the serous pericardium. Sometimes the serous pericardium, this layer right here, is also referred to as the epicardium. They're interchangeable terms. Now, also you're going to hear these terms parietal layer and a visceral layer. I'm going to show you what these look like. So here's the parietal layer, and then the visceral layer of the serous pericardium is also known as the epicardium. And then between those 2 is a pericardial cavity, right in here, the blue layer. Okay, we'll tell you what the importance of that is. So the fibrous pericardium, which is the outermost layer of the pericardium, that's it's primarily tough, inelastic connective tissue. And its primary function is going to protect the heart. It's going to anchor it into its anatomical position. And, because that thing is pumping pretty aggressively, and it's going to prevent the over distension of the heart. So I want to show you the parietal layer and the visceral layer. Remember that the visceral layer is also known as the epicardium. And let me show you through picture here the difference. So here's the heart. Now consider this, think of it as a balloon, right? So as you start pressing that organ into the balloon, you see how it kind of distorts the structure of it? The inside is still the pericardial cavity, so this here is still the pericardial cavity where there's fluid. But now, as that is placed inside, right, it pushes in, it makes this indentation, you're left with something that really appears to have two layers. There's this layer here, and then there's another layer here that's really close to the organ. So the organ is the viscera, right? So that's the visceral layer. The outer part is the parietal layer. So now in that cavity, the pericardial cavity where you have that fluid, the fluid is really important because it's going to dissipate heat from the heart, because that thing is pumping pretty aggressively. Any muscle that's contracting is going to generate heat, right? Think of skeletal muscle. When that thing starts, when your muscles are contracting aggressively, you get hot, you overheat. It's going to lubricate and protect and cushion the heart, and it's also involved in substance exchange. Ions and lactic acid, etc. The myocardium, which is the thickest part of the heart, it's arranged in what's considered to be a functional syncytium or a functional synctium, and we'll show you what that looks like. When you look at the heart under a microscope, we see that heart cells come together and they're joined by, you can see these perpendicular slits there. They're called intercalated discs. Intercalated discs, which is here, it creates something called a gap junction. All right? And gap junctions, if you think back to different types of cells, when they come together, cells make tissues, tissues make organs. So there are different types of cell junctions. There's tight junctions, gap junctions, desmosomes, adherent junctions. So here, gap junctions are always found in muscles for fast, rapid forms of communication. And they're also found in the neurology, in the neural system as well. Look at all the mitochondria that are there. Remember, the heart needs lots of energy. Mitochondria is producing ATP. And you can see that the cardiac muscle does look striated, just like skeletal muscle does. The difference is skeletal muscle is voluntary, and the cardiac muscle is involuntary. Again, another picture just showing how you have these intercalated discs, and then the gap junctions, when they come together, these intercalated discs, just think of them as like two combs. If you take two combs and you interlink them and put them together, the teeth of the combs connect, right? Here's one end of the teeth of a comb and then the teeth of a comb this way. When they come together and you pull them apart, you can see the gap junctions there. And that's going to allow this cardiomyocyte to communicate with this one and to communicate with this one super quick, so that everything is functioning in harmony or in sync, a functional syncytium or synctium. So a functional synctium is that every part, every cell of the heart is recruited at the exact same time, keeping everything contracting in harmony. These electrochemical current that's generated through the heart that starts through the pacemaker at the SA node is going to move very quickly and send its conduction to both atrium at the exact same time, so that the left and right atrium contract in harmony. So that one heart cell of the right atrium is communicating with another heart cell in the left atrium, and they know to contract in harmony. Okay, and I'll go through that with you in another illustration. But, let me just draw something here for you. May help you understand it a little bit. So, if here's the heart, and let me change the color here to red. Now let's say right up here is the pacemaker, and that's called the SA node. And it's going to send a conduction to all cells of the right and left atrium, just like that. So that this little heart cell here and this little heart cell here, they're getting their impulses at the exact same time because of the intercalated discs, because of the gap junctions, so that they're both in harmony. And then eventually it's going to relay it here to the AV node. And then I'll show you in a few minutes how it drives the impulses down in this direction and upward, and why it does that. But both atriums contract at the same time and both ventricles. Atrium, ventricle, and that's going to open and close the valves in a particular pattern, where it's the AV valves and then the semilunars, the AVs and the semilunars, AVs and semilunars. You get this lubbed up, lubbed up, lubbed up, lubbed up sound. All right, so within the atrium, you have these pectinate muscles, and in the ventricles, we have these trabeculocardiae muscles. And in the ventricles, the trabeculocardiae have these hills that I showed you in the video before that are called these papillary muscles that have cords that extend from them upward to the bottom of the AV valves. And the Chordae Tendinae muscles are going to protect the cusps from over closing, or it's going to help prevent them from inverting. So if, let's say here's the heart, let's say here's a valve and here's a valve. And let's say these are the papillary muscles. I'm just going to draw a few here. From the papillary muscles, we're going to have these Chordae Tendinae that are going to go here and attach to the valve. Now, if it didn't, what would happen is, remember, the ventricles contract. When they contract, it's going to drive blood inferior to superior. When that does, this valve right here, if you didn't have the tendons attached, the force of the blood that hits that valve in that direction can slam it not just shut, but it can overshut into this position. It'll open. And if that happens, blood can regurgitate back into the atrium. So having the Chordae Tendinae, it creates a little downward pull, so it just allows enough tension to close it. Okay, that's the importance of the Chordae Tendinae. All right, so much for my drawings, because you can see them a lot nicer over here. Again, you can see the papillary muscles, and you can see the Chordae Tendinae. And you can see how they attach to the valves. Okay. All right, the endocardium consists of endothelial cells. Remember, endothelial cells, they're vulnerable to damage. They can, any type of oxidative stress, high homocysteine levels, poor methylation, which increases homocysteine levels, can damage endothelial, whether it's of the heart or the blood vessels. So since I'm on that, let's just take a look And let's see if we can find, here's the methylation. And if you look here on the left-hand side, you'll see at around the 6 o'clock position of the methionine cycle, you see homocysteine. And homocysteine, think of it in high concentrations, could be like liquid sandpaper to blood vessels in the heart and endothelial, and it could do some damage. So the body can find a way to metabolize that downward into cysteine in the presence of PLP or B6, pyridoxal 5-phosphate, or cysteine. And remember, cysteine and glutamine and glycine, those 3 amino acids, can produce glutathione, a very, very powerful antioxidant for humans. So that's one way that homocysteine can be altered into something safer and more useful for the body. Or homocysteine, when it borrows this methyl group from the folate cycle in the middle, you see the 5-methyl tetrahydrofolate. Well, if it borrows that M, that methyl group, and it combines with homocysteine, it moves upward into methionine. But now you've lost the methyl group, and now you have tetrahydrofolate. Right? I mean, that's what you have, tetrahydrofolate, and tetrahydrofolate has to find a way of getting remethylated as well. Now you have methionine goes to S-adenosyl methionine, to S-adenosyl homocysteine, back to homocysteine. Homocysteine borrows that methyl group and becomes folate. Okay? And now you have homocysteine that borrowed the methyl group from 5-methyl tetrahydrofolate, and it becomes methionine. So here's where, you have this perfect harmony between B6, B9, and B12. So when you have methylcobalamin and methylcobalamin, the methyl group of the methylcobalamin is utilized to convert homocysteine into methionine. Okay, let me see if I could show you another picture. I'm going to borrow it. Let's see if I can find this here. Okay, let me show you something here. Okay, this is really a really nice diagram. It looks a little bit confusing, but I want you to take a look at the lower right where you see homocysteine. All right, now when you see homocysteine here, the middle cycle is a B12. All the way on the left, you have your B9 pathway. So you got B9, B12, and B6 over here on the bottom right, B6, 9, and 12. So you got homocysteine. Now, homocysteine can work its way up and become methionine in the presence of B12 in the form of methylcobalamin. So one of the things that happens is this methyl group is now donated to homocysteine, and homocysteine becomes methionine, just like that other picture showed. Then methionine goes to S-adenosyl methionine, to S-adenosyl homocysteine, to homocysteine. And homocysteine in the presence of B6, or PLP, pyridoxal 5-phosphate, goes to cystathione, which becomes cysteine. Cysteine and glutamine and glycine become glutathione. Now, the methyl group that homocysteine borrowed, now you're left with just cobalamin up at 12 o'clock in the middle. Cobalamin needs to be remethylated to become methylcobalamin. So where is it going to get its methyl group from? That's where the folate pathway comes into place, and you get that 5-methylene tetrahydrofolate. So when cobalamin borrows that methyl group from 5-MTHF, cobalamin becomes methylcobalamin. And what does 5-MTHF become? Tetrahydrofolate. Okay, and now tetrahydrofolate can move in this direction, moving upward to become remethylated as MTHF, but it needs the MTHFR enzyme, the methylene tetrahydrofolate reductase enzyme. This is where those SNPs come into play, where that C677T or the C677T or the A1298C. If you have homozygous or let's say C677T homozygous or A1298C homozygous or heterozygous on both, there's going to be reduction in this MTHFR, which means a reduction in 5-MTHF, which means a reduction or downstream of this entire pathway. All right? So that's the importance of methylation, and this pathway here shows how you need all of this. You need methylation to be taking place because there's a link in keeping all of these cycles happening in order to produce neurotransmitters, right? You got melatonin up here, you got serotonin, you got dopamine, you have norepinephrine, epinephrine, tyrosine, you have thyroid hormones that are produced, so really important process. All right, let's see where we are now. Endocardium. Okay, so we spoke about that. We spoke about the lubbed up. The lubbed up, if you look at the sounds, is always the closing of valves, right? Think of opening a door compared to closing it. Closing it, slamming it shut is what makes the sounds. So the closer of the AV valves, which is the tricuspid and bicuspid, makes the lub, and then the DUP is the semilunars, the aortic semilunar and pulmonary semilunar. So when you look on the left and you look at the right, and we look here on the left-hand side is the pulmonary and the aortic, and then on the right-hand side you have the bicuspid and you have the tricuspid closed. So this is lub, and then this is dup. Lubbed up, lubbed up, lubbed up, lubbed up, lubbed up, lubbed up, lubbed up. So the right closure of the AV valves, atrioventricular, is the lub, and then the dup is the semilunars closing. So the heart sounds, you'll see, oh good, I like this picture. Right here, I'm going to outline this. So here is the manubrium, the top of the sternum, and then here's the body of it, and then here's the xiphoid process, right, the point. So right here is that angle of Louis, right there, that junction. And that's where the second rib is. So then this is the second intercostal space. Third rib, third intercostal space. Fourth rib, fourth intercostal space. Fifth rib, fifth intercostal space. So here are the valves. Pulmonary semilunar, we're not putting the stethoscope right over the valve. We're going to go almost like perpendicular to it, and that's where you're going to end up hearing the sound. It's like an echo. The echoes are always at the opposite end. It's heard louder there. Again, if you look at the tricuspid, you were almost going to go to the apex of the heart to listen to it. When you look at the bicuspid, we're going right there to the apex to hear the sounds loudest. Otherwise, you know, there are these murmurs that can be caused by a stenosis, which is an incomplete opening of the heart valve, a prolapse, where there's an inversion of the heart valve, and the regurgitation is an inversion with significant backflow going from the ventricles up into the atriums. So the chambers of the heart include the two upper atrium and the two lower ventricles. So if you look at the picture here, you can see here's an auricle of the right atrium. Auricle means like it's a little reservoir up here for blood. And then here's the right ventricle. The two chambers of the heart include the two upper atrium, the two lower ventricles. Let's see. The atrium receives blood from the superior vena cava. the inferior vena cava. That's what my video showed. And then there's one other structure that's really important, and that structure is the coronary sinus. Now, the coronary sinus I'll show you in another picture, but there's the opening for it, which is right over here. Let me circle it. Now, what this is that the heart itself is a muscle. Muscle needs oxygen. So the muscle of the heart needs oxygen. And then after the oxygen's been delivered, you have deoxygenated blood. So the deoxygenated blood that's being delivered to the heart muscles itself have to find a way of getting dumped back into the right atrium, just like all the blood being dumped from the superior vena cava and inferior vena cava. All the deoxygenated blood from the brain and the facial muscles and the muscles of the arms from the superior vena cava, and then all the abdominal organ deoxygenated blood and all the blood from the lower extremities have to find a way into the right atrium from the inferior vena cava. The deoxygenated blood from the heart itself coming in from like the great cardiac vein and the middle cardiac vein, from all the veins, have to find a way into this port, this large dilated section called the coronary sinus. And the coronary sinus is then going to dump its deoxygenated blood into the right atrium through the coronary opening of the coronary sinus. Okay, so the right ventricle receives blood from the right atrium, and it's going to then send blood up into the lungs through the pulmonary trunk, right? So you can see going from, let's see if I can change the color to red, because I'm drawing it the blue. So it's going to come this way from the superior vena cava, inferior vena cava. Now it's dumping to the right atrium. Then it's going to pool down into the right ventricle, and from there it's going to open that push valve of the pulmonary trunk into the pulmonary trunk, driving it that way into both pulmonary arteries. Left atrium receives blood from the pulmonary veins. So that's here. Let's make it blue now. So now the left atrium, here's the left atrium, it's going to receive blood from the pulmonary veins. So now you have oxygenated blood coming from the left lung and the right pulmonary veins, bringing blood in from the right lung, also dumping it into the left atrium. The left atrium receives blood from the left atrium receives blood from the left atrium and sends blood all over the body. Let me see what that means. The left atrium is receiving blood from the left. It's got to be from the left and right lungs. Okay? That's it. The left atrium is receiving blood from the left and right lungs, and then it's got to pump it down into the left ventricle. Note that the wall of the left ventricle is much thicker than that of the right. So if you look at this picture, this is a bird's eye view. If you look at the picture, here's the lumen of the left atrium, here's the lumen of the right, but look at the thickness here. Right? Look at the thickness of the muscle. So why? Why is the thickness of that left ventricle so much thicker than the myocardium of the right? Well, think of what the heart is doing. Does it take a lot of energy? Think of it this way. Does it take a lot of energy to pump blood from the heart to the lungs? That's pretty close in proximity, compared to the left side of the heart having to pump blood up to the brain, down to the fingers, down to the toes, down to the organs. So really, the left side of the heart is working much, much harder. If you're only exercising your left arm and doing curls with your left bicep, the left bicep's going to get thicker and stronger than the right bicep. That makes sense, doesn't it? All right, so the valves of the heart open and close in response to the pressure changes. The right and left AV valves are going to prevent backflow from the ventricles into the atrium. If you get backflow, that's called regurgitation. The right and left semilunar valves prevent backflow from the arteries into the ventricles. Okay, we showed you this picture already. You probably want to pause this and print this up and just follow the flow here. This is what my video showed you, but really good to kind of, draw this out yourself. If you look, everything is labeled so beautifully here for you. I also like the keys showing you oxygen-rich blood, so that's oxygenated or deoxygenated blood. Follow that flow. Let's look at this for a second. So you have the heart, systemic arteries, there's your pulmonary, right? So if you look at pulmonary circulation, systemic arteries, hepatic portal, really important concept, the hepatic portal system. We'll go into more detail when we cover the blood vessels a little bit more. But hepatic portal is basically blood that's coming from your digestive tract and your digestive system and a lot of the abdominal organs. They have to find a way of getting blood back into the heart, but it's got to be filtered first. So it's going to send it to the hepatic portal system into the liver, and then after the blood's been filtered and detoxified through phase 1 and phase 2 of liver detox, it's going to drive the blood back into not the hepatic portal vein, but the hepatic vein. The hepatic veins go from the liver into the inferior vena cava. Once it's in the inferior vena cava, it can drive the blood back into the right atrium. And here are the systemic veins. Okay, blood flow through the coronary arteries deliver oxygenated blood and nutrients to the myocardium. Again, the heart is muscle. So let's just review and show you off of the aorta right here, there's one blood vessel coming off of it, and the pulmonary trunk is blocking it, but if this was removed, right here on the other side is another blood vessel, and again I'm going to draw like it's dotted because it's going behind, leading this way into the left coronary artery. So there's a right coronary artery and a left coronary artery. Both of them come off of the ascending aorta, and it's going to bring blood to the myocardium of the heart. Beautiful picture. Now this illustration shows what I was just trying to draw out. Here's the right coronary artery. You can see it's coming off of the ascending. And then here's the left coronary artery, again coming off the ascending aorta. So here's one blood vessel, and then here is the other. So the left coronary, if we follow it down, it's going to branch and go down into the interventricular sulcus, which is kind of like a groove inside on the top of the heart that's going to separate the right and left ventricle. So as that branch or artery comes down, it's the anterior interventricular branch. And then the right coronary artery comes down into the right, into the marginal branch, right down to the apex of the heart. Off of the left coronary, besides the anterior interventricular artery, it's going to branch around to the posterior side. That's the circumflex artery. All right, now the blood has been dumped. Now it's deoxygenated. So you can see we have like the small cardiac vein, anterior cardiac vein, middle cardiac vein, the great cardiac vein. All of these veins are going to dump blood around the posterior side is the coronary sinus. And here is the coronary sinus, which is going to have that opening into the back of the right atrium. All the deoxygenated blood is brought there. Okay, this should look a little bit familiar. It's right, similar arrangement to skeletal muscle. Here's cardiac muscle. And some of the same features. There's the sarcoplasmic reticulum. That's where calcium is stored. We have mitochondria. The transverse tubules is for the action potential to transmit. And that transmission is going to move into the sarcoplasmic reticulum, and the calcium is now released into the thin and thick filaments. Actin is thin, myosin is thicker. The cardiac muscles are the cardiac muscle cells are self-excitable. They're autorhythmic. The cardiac muscle cells repeatedly generate these spontaneous action potentials that trigger the heart contractions, and those action potentials are going down the T tubules, the transverse tubules. So let's show you the conduction system of the heart. So I kind of started drawing this out before, where I showed you the SA node right here, and you could see the arrows going into the right atrium and left atrium. So all of the cardiomyocytes are going to get the impulse, and then it's relayed to number 2, the AV valve, the atrioventricular node. Now here's the trick. Once it's sent to the number 2, the AV node, notice that the pathway, follow the yellow pathway, it's hidden in this wall. This wall is the interventricular septum. The septum is a wall. The interventricular septum is that thick muscular wall that separates the right and left ventricle. Now remember I said how the atriums, when they contract superior to inferior? But when the ventricles contract, they're going to contract inferior to superior. And the only way that happens is the electrical impulses have to hide themselves in the interventricular septum, where now you've got this bundle of His, and the bundle of His then separates into these bundle branches, a left and a right bundle branch. And then these left and right bundle branches divide, they go up the apex, and they move upward inferior to superior to create ththese Purkinje fibers. Now the electrical impulses can make it all the way down to the apex of the heart, work their way up, so that the ventricles can contract inferior to superior. Otherwise, the top part would contract, the top part of the ventricles would contract. And if the top part contracts before the bottom, it pushes the blood down. We want the bottom to contract first and then to fire upward to help support the concept of pushing blood inferior to superior, so that it can drive blood upward and push those push valves open, which is the aortic semilunar and the pulmonary semilunar. Okay, hopefully that makes sense. Let's take a look at the cardiac conduction.
In order for the heart to function properly, cells must contract in a specific sequence. A pathway known as the conduction system determines this sequence. The heart's unique conduction system works because of cardiac muscle cells that are both connective and autorhythmic. Autorhythmic cells spontaneously generate and spread action potentials from fiber to fiber across gap junctions. This triggers the networked heart fibers to contract in a regular rhythm. Cells from different parts of the heart have different natural rhythms, ranging from 100 action potentials, or beats per minute, in the sinoatrial node, to 40 to 50 beats in the atrioventricular node, to only 20 to 40 beats in the ventricles. The SA node is known as the healthy heart's pacemaker. Action potentials generated from the SA node spread to other areas of the conduction system and trigger contraction before the other areas are able to generate action potentials at their own slower rate. As the action potentials move through the atria and reach the AV node, the atria contract, moving blood into the ventricles. Typically, the AV node is the only connection between the atria and the ventricles. As the wave of excitation passes the AV node, it proceeds to the AV bundle and bundle branches, the apex of the ventricular myocardium. The conduction myofibers then directly stimulate the cardiac cells in the ventricles to contract. During ventricular contraction, blood is squeezed out from the apex to the base and into the pulmonary trunk and aorta. The signal moves quickly from the SA node through the atria. It experiences a slight delay when it reaches the AV node, as it must pass into fibers with smaller diameters. The signal then quickly resumes along its path, through the AV bundle and branches, apex, and base of the ventricles. Contraction begins. The sequence of depolarizations and repolarizations can be seen in a normal electrocardiogram, usually called an ECG. The electrocardiogram can also show problems with the conduction system. Look at this trace showing complete heart block. By comparing the QRS waves to the P waves, you can see that ventricular excitation is independent of atrial excitation. The pace of ventricular contraction is being set by the ventricle's slower natural rhythm, not by stimulation from the SA node and atria. A typical contractile cell in the myocardium has a resting membrane potential. Cells in the SA node do not have a fixed resting membrane potential. Instead, the resting membrane potential of an SA node cell is called a pacemaker potential. Each healthy heartbeat starts with a pacemaker potential, in which the autorhythmic cells of the quick-firing SA node spontaneously excite themselves and neighboring cells. The cell's membrane potential gradually becomes more positive until threshold is reached. This triggers rapid depolarization, sending the contraction signal on its way. Repolarization follows, and the SA nodal cell is ready to fire again. In ventricular cells, an action potential has 4 stages. The resting membrane potential is steady until a stimulus causes it to exceed threshold, triggering rapid depolarization, which is followed by the plateau, during which the cell remains depolarized. The plateau is followed by repolarization, after which the cell returns to the resting potential. The SA node's rate of firing, combined with modifications from the autonomic nervous system, determines the heart rate. The autonomic nervous system can modify the heart's rate in 2 ways. The hormone acetylcholine, secreted from parasympathetic neurons, decreases SA node firing to about 75 action potentials per minute, decreasing heart rate. This heart rate is considered normal and healthy for a resting heart. Conversely, norepinephrine increases heart rate. This hormone, secreted from sympathetic neurons, is produced more during exercise, stress, and excitement.
That was a great review right there. Okay, so you can also just kind of go through this flow where it kind of shows the different steps. I just kind of numbered it for you. So we start out at the SA node, which we know is the pacemaker, and it's going to drive that conduction to both atriums so that the atriums contract. And when the atriums contract, it's going to create that little bit of a P wave, and it's certainly not as intense as the QRS wave, because gravity alone is going to help take some of that blood from the atrium into the ventricle. So that's why when you look at the P, Q, R, S, T wave, that this P wave here is not as intense as the Q, R, S wave. There's the P, Q, R, S, T wave. So right in here, that's atrial depolarization or atrial systole. Okay, and then it's going to, the atrium is going, the SA node is going to relay that information to the AV node, and then from the AV node to the bundle of His, and then the bundle of His is going to divide into the right and left bundle branches in the interventricular septum, and then those bundle branches move downwards to the apex, up and around to the right and left ventricles, where they divide into those Purkinje fibers. Okay, and then when those ventricles contract, that's ventricular depolarization or ventricular systole. That's the QRS wave that really does spike quite a bit. When the ventricles are in contracting, that's ventricular systole. That means that the atrium have to be resting, or they're repolarizing. They're resting or they're repolarizing. That's diastole as well. When the atriums are contracting, the ventricles are resting or repolarizing. That's diastole. Okay, so think of systolic as active contraction, and repolarization is more relaxation. So systole is contraction, diastole is relaxation. Here, nice diagram showing the P wave. There's your atrial depolarization. Then you can see this spike of the QRS, and here, the QRS, that's ventricular depolarization. And then that little T is when the ventricles are now relaxing for the T wave. Now, if there's any type of abnormal findings, where if you look on the left there, here's your P, Q, R, S, T wave. When you start to see problems like, look at this T, and let's compare it to this T here. So you see you have an inverted T wave. And then what's happening here between, right here, you have an elevated ST segment. And then here, look at that Q compared to this Q, you have a significant Q wave. So these are all abnormal findings, and they're all kind of working their way towards a heart attack, essentially. You know, it starts off with, you know, lack of blood flow, ischemia, then the muscle is going to get slowly injured, and then eventually you get that infarct, or the MI, which is a myocardial infarct. And that's what it ends up looking like, dead black tissue. That's a myocardial infarction. This is a person that's going to experience angina, you know, angina pectoris, which is that chest pain. That's when they have to put, you know, nitroglycerin underneath the tongue, which is really just nitric oxide to get the blood vessels to dilate, to try and drive blood into the heart. The autorhythmic fibers in the SA node are the natural pacemaker of the heart, because they initiate the action potentials most frequently, and signals from the neural system and hormones like epinephrine can modify the heart rate and the force of contraction, but they do not set the fundamental rhythm. The heart muscles generate ATP via anaerobic cellular respiration and creatine phosphate, or CP, creatine phosphate. Here's the action potential propagation. You can kind of go through this yourself, but it just kind of shows what that other illustration was showing, how it had the overlap between what the heart was doing and what the valves were doing, and whether it was systolic or diastolic. So really nice to kind of show it. And it does go in order if you kind of follow the sequence, where you can start with number 1 in here and then just work your way around. Okay, so it's showing you what's happening, You can see the QRS tells you what's happening. So take some time, and even the picture shows you how the blood is pooling up in the lungs and in the heart at different levels. One cardiac cycle consists of the contraction, systole, and relaxation, diastole, in both atria, rapidly following by systole and diastole of the ventricles. There's electrical events, there's pressure changes that happen, there's heart sounds, volume changes, and mechanical events. Let's look at the cardiac cycle here.
The heart beats about 100,000 times each day. One heartbeat consists of a cycle of events that lasts about 0.8 of a second. Each cycle includes the contraction and relaxation of both the atria and the ventricles. In each chamber of the heart, the sequence of activity begins with excitation. Excitation causes contraction. Contraction increases blood pressure. Changes in pressure cause the opening and closing of heart valves, regulating the volume and flow of blood. Timing of the sequence depends on the amount of electrical activity in the heart muscle. Electrical changes during the cardiac cycle can be seen on an electrocardiogram, or ECG. Electrodes placed on the skin detect changes in the heart's electrical activity. The P wave indicates atrial excitation. The QRS wave indicates ventricular excitation. The T wave indicates the end of ventricular excitation. Atrial contraction, or systole, initiates the cardiac cycle. Phases that follow are isovolumetric contraction, ventricular ejection, isovolumetric relaxation, and ventricular filling. Prior to atrial contraction, the atria and ventricles are relaxed. Atrioventricular valves are open, and semilunar valves are closed. The ventricles fill with blood. Depolarization triggers atrial contraction. Contraction increases atrial pressure and pushes extra blood into the ventricles, slightly increasing ventricular pressure. The excitation triggers ventricular contraction. Ventricular contraction is at first isovolumetric. This means that ventricular blood volume and muscle cell length do not change. Contraction causes ventricular pressure to rise above atrial pressure, forcing the AV valves shut. With the valves closed, the continuing contraction increases ventricular pressure. Ventricular ejection takes place when ventricular pressure rises above arterial pressure and the semilunar valves open. The valves stay open for about a quarter second. As blood is ejected into the arteries, ventricular volume decreases. Ventricles begin to repolarize and relax, ventricular pressure decreases, and contraction ends. Repolarization of the ventricular muscle cells initiates isovolumetric relaxation. As the ventricles relax, pressure in the ventricles drops. Semilunar valves close, preventing backflow. The closure produces a dicrotic wave on the aortic pressure curve. With both semilunar and AV valves closed, blood volume does not change. This isovolumetric relaxation sees pressure drop quickly. Ventricular filling occurs as all four chambers of the heart are relaxed and the AV valves open. As the valves open, the ventricles begin filling with blood. In about 0.2 of a second, blood that has been building up in the atria rushes in to fill the ventricles. Atrial depolarization triggers atrial contraction. forcing a small amount of extra blood into the ventricles. And a new cardiac cycle begins. There are two major heart sounds that make up a heartbeat. They are known as S1 and S2. The heart sounds result from audible blood turbulences of blood flow caused by the closing of heart valves. The S1 sound is described as a lub sound. It's louder and lasts a bit longer than the S2 sound. S1 is produced by the closure of AV valves and marks the start of ventricular contraction. The S2 sound is described as a dup. Its pitch is higher and sharper than S1. S2 is produced by the closure of SL valves and marks the start of ventricular relaxation. The sounds together form the heartbeat and sound like lubbed up.
Okay, let's look at the cardiac cycle real quick.
The constant circulation of blood throughout the body is critical to life. It is the pumping of the heart that provides the force needed to move blood through 60 to 100,000 miles of blood vessels, thus allowing for the exchange of gases, nutrients, and wastes for the trillions of cells in our bodies. The heart acts as two side-by-side pumps. The right side, responsible for pulmonary circulation, pumps oxygen-poor blood from the body to the lungs. The relatively lower pressure in the right side of the heart protects the delicate tissue of the lungs and allows the thinnest possible barrier between the air in the lungs and the capillaries. At the same time, the left side pumps oxygen-rich blood at higher pressure to the body via systemic circulation. The heart has four distinct chambers. The upper two chambers, the atria, constantly receive blood. The lower two chambers, the ventricles, function as pumps to provide the force needed to push blood through the system of vessels circulating throughout the body. The direction of blood flow through the heart is maintained by four one-way valves. The atrioventricular valves prevent blood from flowing back into the atria during ventricular contraction. The semilunar valves prevent the flow of blood back from the pulmonary trunk and aorta into the relaxed ventricles. When heart valves close, vibrations occur within the ventricles and blood vessels that can be heard through a stethoscope. The cardiac cycle has 5 phases and includes all of the events that occur during a single normal heartbeat. Phase 1 is passive ventricular filling. All of the chambers are relaxed, as most of the blood from each atrium passively flows through the open atrioventricular valve into its respective ventricle. Atrial contraction is the second phase of the cycle. The atria contract, forcing additional blood into the ventricles. Atrial contraction causes ventricular blood volume to reach its maximum. Phase 3, isovolumic ventricular contraction, occurs as the ventricles begin to contract. Pressure rapidly increases within each ventricle, forcing the atrioventricular valves to close. Since both atrioventricular valves and both semilunar valves are closed, blood pressure within the ventricles increases, but the blood volume actually remains unchanged. This is said to be isovolumetric. Ventricular ejection is the fourth phase of the cardiac cycle. As ventricular contraction continues, the pressure inside the ventricles eventually exceeds the back pressure in the pulmonary trunk and aorta. This forces the semilunar valves to open, and blood from the ventricles is pushed into these great vessels. In a resting adult, slightly more than half of the blood, approximately 70 milliliters, is ejected each time the ventricles contract. The combined third and fourth phases of the cardiac cycle, when the ventricles are contracting, are known as systole. The final phase, isovolumetric ventricular relaxation, occurs when the ventricles begin to relax. When the pressure in the ventricles becomes less than in the great vessels, the back pressure pushes the semilunar valves closed. Because all four valves are again closed as the ventricles relax, there is no change in blood volume, causing a second isostate, known as isovolumetric ventricular relaxation. From this phase until the start of the next ventricular contraction is known as diastole. Each cardiac cycle lasts about 0.8 seconds, a resting adult heart rate of approximately 75 cycles per minute. Even though there is continual contraction throughout life, this muscle never tires because the ventricles are in systole for only 0.3 seconds and diastole for approximately 0.5 seconds. The ability to rest for more than half of the cardiac cycle is sufficient to maintain normal heart function. During strenuous activity, like exercise, the heart has the ability to increase its output by 4 to 5 times. It is an organ of amazing design and capacity.
Okay, let's look at the EKG, the P wave, the QRS, and the T wave.
The heart beats about 100,000 times each day. One heartbeat consists of a cycle of events that lasts about 0.8 of a second. Each cycle includes the contraction and relaxation of both the atria and the ventricles. In each chamber of the heart, the sequence of activity begins with excitation. Excitation causes contraction. Contraction increases blood pressure. Changes in pressure cause the opening and closing of heart valves, regulating the volume and flow of blood. Timing of the sequence depends on the amount of electrical activity in the heart muscle. Electrical changes during the cardiac cycle can be seen on an electrocardiogram, or ECG. Electrodes placed on the skin detect changes in the heart's electrical activity. The P wave indicates atrial excitation. The QRS wave indicates ventricular excitation. The T wave indicates the end of ventricular excitation.
Good summary. Those last two videos were really good summaries. Okay, cardiac output is the volume of blood that's ejected from the left or the right ventricle into the aorta or the pulmonary trunk each minute. Stroke volume is the amount of blood pumped out of the ventricle in one beat. Okay, and there's a little formula down below. Cardiac output equals stroke volume times the heart rate. Let's look at cardiac output.
The pumping action of the heart maintains blood flow throughout the human body. Cardiac output is a measure of blood flow. It is the volume of blood ejected from the heart over a given time. Cardiac output is determined by multiplying heart rate by stroke volume. Cardiac output in the typical resting heart is about 5 liters per minute. Cardiac output is not constant. It varies with the body's metabolic demands. Mild exercise will double cardiac output, while an intense workout can increase output as much as 4-fold to 20 liters of blood each minute. Stroke volume is directly correlated with cardiac output. The greater the stroke volume, the greater the cardiac output. Stroke volume represents the difference in the amount of blood between the volume in the ventricles at the end of diastole and the volume after systole. Three factors increase stroke volume: increased preload, increased contractility, and decreased afterload. preload is the degree of stretch of cardiac muscle cells prior to contraction. The amount of stretch is related to the end diastolic volume. Increased return blood flow from the veins increases end diastolic volume. Cardiac muscle sarcomeres stretch and lengthen. Contraction of the cardiac muscle sarcomeres during ventricular systole provides the force to eject blood from the heart. Sarcomere length affects muscle tension and the force of contraction. An increase in muscle stretch increases tension. A more forceful contraction ejects more blood, thus increasing stroke volume. The relationship between preload stretch and stroke volume is known as the Frank-Starling law of the heart. The greater the preload, the greater the sarcomere stretch, the greater the stroke volume. The Frank-Starling law has limits. When cardiac muscle is overstretched, increasing ventricular filling has no significant effect on stroke volume. Contractility is the forcefulness of cardiac muscle contraction. Inotropic agents can increase or decrease contractility at any given level of stretch. Positive inotropic agents increase contractility, thereby increasing stroke volume and cardiac output. Negative inotropic agents reduce force of contraction. Positive inotropic agents include neurotransmitters, hormones, and some drugs. Certain drugs, such as calcium channel blockers and some anesthetics, are negative inotropic agents and reduce contractility. Afterload is the pressure or force that the ventricles must overcome to push open the semilunar valves and eject blood. Blood pressure in the left ventricle must overcome arterial pressure in the aorta in order to open the aortic valve. Hypertension increases aortic pressure, and the left ventricle must work harder to generate the pressure required to maintain stroke volume and cardiac output. Eventually, ventricular pressure builds to overcome the elevated arterial blood pressure, and blood is ejected from the heart. As afterload increases, ejection is more difficult, and both stroke volume and cardiac output are diminished. The ventricles compensate for decreased cardiac output in two ways: by activating feedback mechanisms that increase preload, and by initiating hypertrophy in cardiac muscle cells to increase contractility. The sinoatrial node normally initiates 60 to 100 heartbeats per minute. A normal resting heart rate is about 75 beats per minute. Heart rate and cardiac output are directly proportional to one another. The greater the heart rate, the greater the cardiac output. Heart rate is influenced by such conditions as exercise, stress, blood loss, and tissue damage. More importantly, the autonomic nervous system and hormones play key roles in the regulation of heart rate. A decrease in blood pressure promotes sympathetic stimulation, the release of norepinephrine, and thus an increase in heart rate and cardiac output. An increase in blood pressure promotes parasympathetic stimulation, the release of acetylcholine, resulting in a decrease in heart rate and cardiac output. The hormones epinephrine and norepinephrine, secreted from the adrenal gland, as well as thyroxine, accelerate heart rate. Low blood oxygen and pH, as well as high blood carbon dioxide, also trigger an increased heart rate. Additionally, moderate increases in cytosolic levels of ionic calcium also increase heart rate.
All right, so those three factors that the video went over was the preload, the contractility, and the afterload. And this is just a really nice picture and illustration that just reviews that, okay? You can see here, like up on top, for example, where it talks about positive ionotropic agents, such as increased sympathetic stimulation, catecholamines, remember norepinephrine and epinephrine, etc. They increase calcium in the extracellular fluid, so you get that increased contractility. You get that increased stroke volume. You get increased cardiac output. Okay. Several factors that regulate heart rate: the ANS, the autonomic neural system, hormones, ions, age, gender, physical fitness, and temperature. Let's look at the regulation.
The autonomic nervous system can modify the heart's rate in two ways. The hormone acetylcholine, secreted from parasympathetic neurons, decreases SA node firing to about 75 action potentials per minute, decreasing heart rate. This heart rate is considered normal and healthy for a resting heart. Conversely, norepinephrine increases heart rate. This hormone, secreted from sympathetic neurons, is produced more during exercise, stress, and excitement.
So when you look at certain regulators, it's really important to understand the major influence, especially in that region right here called the medulla oblongata. Remember the importance of the vagal nerve, cranial nerve number 10, but in the medulla is the cardiac center, the cardiovascular center, really important. There's lots of research out there showing how the integrity of the top two vertebrae, let's say C1 and C2, your atlas and your axis vertebra, any subluxation or any type of subluxation or interference to this area can affect this. I've seen it personally in practice over the last 23 years, how I could do a pre-adjustment check on my patients, get their blood pressure, and then make a specific adjustment to their cervical spine, the upper cervical, C1, C2, let them rest, post-adjustment, take their blood pressure again, and watch significant changes to that. So really important area, that medulla. Remember the cranial nerves, 8, 9, 10, 11, and 12 are located there. The nucleuses for those lie there. The input to the cardiovascular center from the higher brain centers, the cerebral cortex, the limbic system, the hypothalamus. There are sensory receptors, proprioceptors that are going to monitor the movement, chemoreceptors that monitor the blood chemistry, and baroreceptors for blood pressure. They monitor the blood pressure, especially in the carotid sinus. There are baroreceptors there. Regular aerobic exercise can increase cardiac output, can increase your high-density lipoproteins, decrease your triglycerides, improve lung volume, decrease blood pressure, and certainly assist in weight control, because when you put exercise, you tend to put on more muscle, more mitochondria. It's called mitochondrial biogenesis. More mitochondria, the more little burning centers you have to burn fuel, which loves to burn fat cells. Okay.
Cardiac output is not constant. It varies with the body's metabolic demands. Mild exercise will double cardiac output, while an intense workout can increase output as much as fourfold, to 20 liters of blood each minute.
In terms of development of the heart, I just wanted to show you how during the initial stages of heart development, you really don't see the distinct 4 chambers, but it starts to, you see here, there's no division separating both of those atriums. And as it starts to form, you can see that there's a foramen ovale, there's a hole here. And many times you'll hear of adults that are going to have a procedure to have a hole fixed in their heart. Usually it's trying to close this up, okay? Sometimes there are developmental issues in where this doesn't formally fuse up and close. There's coronary artery disease that's common. There's atherosclerotic plaques that take place. Congenital heart defects, arrhythmias, congestive heart failure, all different things that can go on in terms of imbalances in the cardiovascular system. When we get into the blood vessels, we'll show you a little bit more what these plaques look like and how blood pressure goes up as a result of it. But here it shows when you have these imbalances, when you have some sort of stimulus that's rising in blood pressure, they are the baroreceptors in the carotid sinus. They send these impulses that travel from the baroreceptors to the inhibitory center. And as a result, it's going to decrease sympathetic impulses to the heart, which will decrease the heart rate, decrease contractility, and create vasodilation where the blood vessel diameter increases, which will decrease the cardiac output, decreases the resistance, right? Whenever you vasodilate and the smooth muscle of the blood vessels open up, you have decreased resistance and blood pressure goes back to normal. If the blood pressure dips and the same baroreceptors pick that up, then it's going to stimulate the vasomotor center, increase sympathetic tone, increase heart rate and contractility, vasoconstrict so that the resistance goes up, the peripheral resistance increases, cardiac output increases, and blood pressure also goes up. Some of the hormones like norepinephrine and epinephrine can increase blood pressure. Those catecholamines are released from the adrenal medulla. In the adrenal cortex, that's where you have cortisol, which also does the same thing. It increases blood pressure. Antidiuretic hormone, ADH, causes intense vasoconstriction. And when you have vasoconstriction of blood vessels, the pressure goes up. Angiotensin 2 comes from angiotensin 1. You need the angiotensin converting enzyme to go from angiotensin 1 to angiotensin 2. Remember, the kidneys release renin, and renin is going to convert angiotensinogen, which is produced by the liver, into angiotensin 1. Then angiotensin 1 is converted to angiotensin 2 because the lungs make angiotensin converting enzyme. And then angiotensin 2 increases blood pressure by vasoconstricting the vessels, releasing ADH, releasing aldosterone. When you have aldosterone, you are going to hold on to sodium. And when you hold on to sodium and minerals, water retention takes place. and then you become, you also increase salt cravings, and you become thirstier. All these things are going to increase blood pressure. ANP, atrionatriuretic peptide, that's your like antagonist to aldosterone, right? So atrionatriuretic peptide is going to release sodium. When you lose sodium, you lose water, and that's going to help pressure decline. NO, nitric oxide, that's a vasodilator. Then you have some chemicals like histamine and prostacyclines. These thin the blood and vasodilate as well, which also lower blood pressure. And then alcohol causes blood pressure to drop by inhibiting ADH, right? If an antidiuretic holds on to water and you block it, you're losing water. Okay, we'll go over this. I think it would be better to show you in an illustration or a picture how all these things work. So here you go. So we have decreased pressure. If we start up at the top here, you got decreased pressure. So what are the kidneys going to do? They're going to release renin. And what's renin going to do? Renin is going to take that angiotensinogen that's produced by the liver and convert it into angiotensin 1. And then the lungs are going to convert angiotensin 1 into angiotensin 2. And that's zinc dependent. Angiotensin converting enzyme is zinc dependent. Then angiotensin 2, we spoke about aldosterone, and that's a mineral corticoid that's released by the adrenal cortex. Angiotensin 2 is also involved with antidiuretic hormone. ADH is produced by the hypothalamus and stored in the posterior pituitary. That's going to increase water absorption, whereas aldosterone is going to increase sodium reabsorption. We know that salt and water always kind of follow each other. They go together. This way, you have an increase in blood volume, and you're going to increase blood pressure. Angiotensin 2 also vasoconstricts. Okay. Just wanted to show you some of the areas where pulses can be palpated and felt. There is the temporal artery, really important area, FYI, just especially in smokers. People today still smoke regardless. If they have these temporal headaches or are very sensitive to pain along that temporal artery region, have to consider temporal arteritis. Pretty serious condition, can lead to blindness. There is the common carotid artery right along the SCM muscle, right in here. Blood pressure is taken through the brachial artery, right there. And then you have the radial artery for the radial pulse here. Femoral artery, behind the knee, popliteal artery, posterior tibial, and the dorsal pedal. A lot of times with erectile dysfunction, they'll check to see how the blood supply is in the lower extremity. Oftentimes when I'm doing my physical exam, I like to feel the dorsal pedal. I want to see, can I feel a pulse here? If not, could that be why they're getting cold toes? How is the circulation below the popliteal artery? Could that be a reason why there's lack of hair to the lower portion of the extremity of the leg? Right? Sometimes that lack of hair is thyroid. Sometimes it's just poor circulation. When we talk about blood pressure be less than 120 over 80. Blood pressure is an inflammatory biomarker. We just don't look at blood pressure as like a heart problem or a blood vessel problem, but it's an indicator. It's a biomarker for inflammation through the body, and trying to find out where the source of inflammation is coming from is the key. So the blood pressure of the vessels when the heart is beating, the systolic number, the top number, we want less than 120. The pressure between the beats is diastole. That's when the heart is relaxing or the ventricles are relaxing. We want that pressure to be less than 80 millimeters of mercury. 120 over 80 or less than that. That's normal. If it's between 120 to 139 over 80 to 89, that's pre-hypertension. And if it's 140 over 90 or above, that's high blood pressure. Okay, so there was a lot of information, but I'm sure you learned quite a bit.